also online this week. So there are a lot of difficulties there too. All right, so I'm just going to introduce myself. My name is Connie Chang, and I'm an assistant professor here in chemical and biological engineering at Montana State University. So my lab studies microfluidics, which I'll talk to you about, and we also study viruses. So in my lab, we study influenza, and recently, since the pandemic happened, we've been studying SARS coronavirus, SARS-CoV-2 here. Okay, so you guys know all about the pandemic. We're living in it now. I like to say we're living in an experiment. Coronaviruses, the reason why they're called coronaviruses is because it's a virus that has an RNA inside, right? It's a single-stranded RNA, but on the outside is a capsid. And that capsid looks very much like a crown when you look at it from the side. So you can see here an image of SARS-CoV-2. You can see that the RNA is on the inside. And you have all of these proteins kind of in the capsid shell on the outside. Let's see, so it's the cause of the common cold. I don't know if you guys knew that, but it's pretty common. And then you sometimes have these more virulent strains or more dangerous strains, such as MERS, which came out from camels in 2012. And also the first SARS, which came out in 2003. Now, in December of 2019 is when SARS-CoV-2 happened. And you know, they had a hard time naming it, right? Because they didn't want to name it after, say, after a country, such as like you think about the 1918 influenza virus, which is often called the Spanish influenza, right? Spanish flu. So they came up with the name SARS-CoV-2. And then they called the disease COVID-19. I often think that they should have just called it SARS-2, which is what it is, but you know, they have this long name, SARS-CoV-2. Okay. Okay. Have you guys gotten this test? Okay, so this is how you would test if you have COVID-19. It's typically using a nasopharyngeal swab, which is a 6-inch long swab, and it goes into the cavity between your nose and mouth. So you can see actually how far it goes. I'm going to show you this video, which is from the New England Journal of Medicine. And basically, oh, we do need volume here, but she sniffs into a tissue. The doctor tilts her head back. This long, it's actually quite flexible, swab goes really deep into the back of the nasal pharynx. And then the doctor twists it a few times and then pulls it out. So hopefully you won't have to experience that too often. But that is how you typically get tested, and we call that the CDC assay, the current assay. So let me tell you a little bit about the swabs. A lot of people, including Jim Wilking here on campus, are 3D printing them. So this is a particular 3D printing company called Form Labs that's actually based in Somerville, Massachusetts. And you know, they're printing 100,000 swabs per day in collaboration with USF Health and Northwell Health. And you can see these, you can get this particular resin that is autoclavable, which means you can make these sterile. And here you can see, yeah, I don't know what happened to my laser pointer, so it's, I'll just use the computer. Okay, so here you can see, do you guys know how the 3D printer works? Can you see this?
No, I can't see our mouse.
They can't see my mouse. All right. So anyway, you have a resin, and it is autoclave. You shine light onto that resin, and that light basically cross-links your resin so that you get a solid material. And then as you 3D print, the whole top, the print head lifts up so that you slowly print these long tubes. And so you can see them here on the left. And here's the package that it actually can come in on the right. All right, so you need a trained personnel to do the nasopharyngeal swab. Nowadays, you'll see a lot in the news about self-swabbing in the anterior, kind of just the lower part of your nose. But what's considered the gold standard CDC test is based upon this swab, where you may go through a drive-through, or you may go into the 10th that's on 7th. And you see the swab. Now the swab is then put into a tube of liquid. So what is that tube of liquid? It's called viral transport media, and it's this is the recipe for it. It's a very typical media that we would use in a laboratory, in a viral laboratory. You can see here that Ryan Langelas, who's an influenza virologist at University of Minnesota, has made a whole lot of them in his lab. So he has a refrigerator full of these 15 milliliter conical tubes with 3 mils of viral transport in each one. Okay, so you take the swab, you put it in the media, you send that tube off to be analyzed. So how do you analyze it? Well, this is a very, very typical method that we use in my laboratory practically every day. It's called qPCR. So the Q stands for quantitative PCR, and the RT stands for reverse transcription. So with RNA viruses, what you need to do is you need to first convert the RNA to DNA, and then you can use polymerase chain reaction to amplify the RNA into many, many, many copies of itself, of more RNA. Oh, okay, so like I said, you have to somehow access that RNA. So remember I showed you the video of the virus particle in the very first slide? Well, you have that RNA on the inside. And the trick is to access it so that you can amplify for it. So somehow you have to break open the capsid. You can do what we typically do in the lab, which is called RNA extraction. And we use these kind of small spin centrifuge tubes. And we just do a few washes. It's actually quite laborious, but maybe takes about half an hour. And the thing about the RNA extraction is that these little kits to isolate the RNA are a little bit pricey. So I say it takes about an hour with all of the kind of pipetting and things like that. Half an hour to an hour. Okay, so next you're going to fill in what we call a 96 well plate. So you have, each well is going to be a different assay, and so you're going to fill up this entire well with PCR mix, which is just a mix that has an enzyme in there to amplify for the RNA, and some primers, which I'll talk about, which help the enzyme know exactly where to amplify on the RNA strand. So you fill this up. You might have a robot to do it. And then finally, you stick it into this qPCR machine, this quantitative real-time PCR machine. And what it does is it starts to amplify so that your enzyme will turn it from a RNA to a DNA, and then your PCR machine will start to amplify this DNA. And then you look at your result. So as you can see here, if you're positive for COVID, you will see this curve start to amplify. And so maybe you can't see the axes, but on the x-axis, it says cycle number. And
I'll go back to the meeting window.
Section.
Okay, yeah, somehow, yeah.
This one, right?
Yeah.
No. This other one. Okay. Interesting. Okay, so if you're positive, you'll see an increase in signal, and on the x-axis here, you have your copy number. So that's the number of times you're thermocycling from a low temperature to a high temperature to replicate the RNA. And if you're negative, you won't see that signal. So a little bit more in detail from the molecular biology perspective. Here you have your target RNA. You have what's called primers. Remember I said they attach onto certain parts of the RNA. You have a forward and a reverse. So first the enzyme will do reverse transcription, which turns it into cDNA. And then you denature it through high temperatures. So you bring this up to 95 degrees for 15 seconds, and your DNA turns into 2 strands of RNA. Then you add on these primers, so you have a forward and the reverse primers. There's these little blue bits here. And you have what's called a Tachman probe. That's pretty common in qPCR, so it's called a Tach reaction. Actually, Tach is an enzyme that was found here in Yellowstone National Park. So that was really neat. Some researchers here do sampling, which you'll hear about later, and I got to go with them once and go see the pool that Tach polymerase came from. So then you take these primers. The primers know exactly where on the RNA to amplify. And then you have all of these different bases that will come on to make the RNA. So you notice that that little bit came off, this black bit. That bit is a fluorescence probe. We call that the Tachman probe, and we measure the fluorescence of that depending on, so it comes off when you make with DNA. So looking at the cycle numbers, you'll see that the fluorescence starts to increase over time, and the neat thing about qPCR is they call it quantitative because you can figure out the amount of RNA that is in your sample. So if you have high amounts of RNA, your curves will start to amplify really quickly at low cycle numbers. So this one is starting to amplify maybe around 10. This is pretty high amount of virus you have in there. And so then what you do is you can kind of plot this line down here, we call that the cycle threshold. And as you go to lower and lower amounts of RNA, this cycle threshold starts to increase. So here you can see we're maybe at 18, 20, etc. And so the higher the cycle number, the lower the amount of virus you have. And then we can plot that, and we can plot kind of an efficiency of your standard curve. And that efficiency you want somewhere between 90 to 110%. So you guys will see in the news a lot where they talk about infectious virus, right? And the thing about qPCR is it's really sensitive. It's sensitive down to, say, 1 virus in 1 microliter, which is a really, really tiny, tiny volume. So it's really sensitive. So the CDC assay says you're positive as you cycle. If you see your curve go up like that at any of the cycle numbers between 0 and 40, which is ridiculous because you'll also read in the news that, there's this importance of rapid testing, right? So when you're looking at the really high cycle numbers, you have very little virus there that is likely not infectious. So basically, the CDC assay is diagnosing a lot of people and saying, yes, you're positive for coronavirus when you could just be recovered from coronavirus and have lingering amounts of virus that's detected through this really sensitive assay called qPCR. Okay, so we do this routinely in my laboratory, like I said, and so we have actually a BSL-3 facility here on campus. So this is kind of a high containment facility where we work with this virus. A lot of viruses that come out that are emerging will first go into either kind of a higher level. There's 4 levels of biosafety, 1 to 4. So in my lab, where you just walk in, we generally work with BSL 1 and 2. So influenza is BSL-2, for example. If you're immunocompromised, it can be a problem, but generally, you'll get a flu shot. Generally, you'll recover from influenza. So that's considered BSL-2. Well, these emerging viruses go into BSL-3, which is high containment, and then BSL-4, which is even higher containment, where you're kind of wearing an entire suit to work with the virus. So we have one here at MSU, which is really neat. And several researchers, including my lab, are in there every day working with the coronavirus. And we've extracted the virus and then brought it over to my lab, and we ran this qPCR. So here you can see we did dilutions of our virus, and we are looking at 2 different segments, the RDRP and the E. And both of those were in the capsid that I showed you in the first slide. So yes, so we have nice standard curves here, and that's really typical. And we need to do this. This is like one of the very first things we did in March, because it's a way to quantify virus, right? So you can see here, if you look at the viruses that are coming out from these infected vero cells, they're probably somewhere at like 10 to the 8 copy numbers. So that's quite a lot. Okay, so this is what I was talking about when you're reading in the news about infectivity and kind of this push for rapid testing, right? So I don't know if you, I don't know how long it's taking now to get tests. Does anyone know, to get your test results back? 2 days, maybe? Well, the turnaround time in which you get your result is really important. And if you look, say like you go to the doctor at, if you go to the doctor too late, those 2 days can really make a difference in terms of infecting other people. So this is the whole thing when I was talking to you about qPCR and CT values. If you look here, this is essentially the viral load and the days since exposure. So really, you're really transmissible in these really high viral loads. And so what qPCR is testing is you can test all the way down to, I would say, 10 to the 1 viral load. That's just about like what I said, 1 copy per microliter. Or 10 copies per microliter. But the thing is, it's almost more important to have a really rapid test that can detect the virus at any of these points, so that your turnaround time for results can be really quick. And like I said, qPCR can measure virus here when you've been, exposed to the coronavirus and it's been over 2 weeks, but your body's still shedding the virus. So in that case, you're still considered positive, but you're no longer transmissible. Okay, so how do we do some of this rapid testing? Well, my lab has been exploring what we call loop LAMP, which is loop-mediated isothermal amplification. So like I said, the qPCR takes about 2 hours, right? You're thermocycling between the 65 degrees and 95 degrees over 40 cycles. This was invented back in 1985. It is slow. It's also expensive. The CDC test is probably about $25 per test, and hospitals are charging you anywhere between, I don't know, maybe $100 to, the astronomical prices that you're seeing. But the other thing is it's tied to a lab, right? So you have to do this by somebody who's trained in PCR. And then in contrast, this loop-mediated isothermal amplification, LAMP, was invented about 20 years ago in 2000. And it is actually, a lot of people, it's fairly well studied. I guess not to the extent that it has been recently, since this past year. And the reason why is because it is really rapid. So LAMP amplifies anywhere between like 5 minutes to 15 minutes. The whole test takes probably 30 minutes if you just want to make sure you don't, to look at all the low levels of virus that you might have amplifying. And it's rapid, like I said, under 30 minutes. It's really cheap. It's probably $1 an assay. And it uses really basic equipment. So it's isothermal, which means it's steady at one temperature, which means all you need is something at between 60 to 65 degrees, which means you can, you know, I have a friend who did this in a coffee cup that he put hot water in. We do it in our lab. We've done it in an oven. We've done it in like kind of a heat block. We've done it in a PCR machine. And all you need is really kind of water at 65 degrees. So single temperature, it's very field deployable, and it's robust. So I'll show you why it's field deployable in a bit. Okay, so LAMP really allows you to do very, very rapid screening, right? Because the test is so fast. You get your results, I would say, within half an hour. And it can be a way to test, you know, asymptomatic people. So one of my grad students this summer thought she was exposed, you know, quarantined herself for 2 weeks, and then was negative. She got her test, and then, you know, her test didn't come back for like 2 weeks. So by the time she got her test results back, you know, 1, she was negative, and 2, she had already quarantined herself for 2 weeks. So that's where this rapid test would have been very useful. It's cheap, it's fast, and it's colorimetric. So you can see by eye whether or not you have the virus. So if it's negative, if it's pink, that's negative. And these guys all have influenza or coronavirus in there. So they're positive and they're yellow. Okay. A lot of different universities are using it. Here on the right, you can see the University of Wisconsin has been using it to test their schools. So they're going out to the schools. They call it a pop-up van. And they're going to the schools and they're testing, you know, all the students at a local elementary school here. I'll show you how the test works, but you know, it's really simple. Currently, you do need to have somebody there to do one pipette step, but LAMP is being innovated to make these promising point of care diagnostics. So that's kind of my area of research. I'll tell you a little bit more about that, but point of care diagnostics such as paper microfluidics, lateral flow devices, LAMP-seq, sequencing is a big part. You'll amplify the virus first and then sequence it. And let's see. Yes, again, it's used commonly in field testing of viruses. So it's been used for Zika, Ebola, dengue, but no one has really used it for kind of clinical diagnostics, right? So that's why there's been a ton of research this past year on how to make this even more sensitive and specific. Okay, so my idea is saliva. I think you guys have been seeing there's been a lot of what they call rapid saliva tests. The rapid saliva tests that have been approved by the FDA through the emergency use authorization, the 2 places where that came out was Yale was one and UIUC was the other. They call it rapid, but it's still tied to a lab. It's still qPCR, so it's still 2 hours, right? But it's still a lot faster because you take away that RNA extraction step, which is 1, expensive, and 2, it you know, adds an extra hour of processing. So saliva is pretty great. So you don't have to have an uncomfortable swab. You know, it's easy to spit into a tube. You don't have coughing and sneezing when you get that swab shoved up your nose. And you can go directly to analysis just from heating your saliva a little bit. So remember how I told you have to access the RNA to do qPCR? Well, you can use heat to access the RNA. And so that's what's done here. Both by Yale and UIUC, you heat up a saliva sample up to close to boiling at 95 degrees for just a few minutes. And then that's it. Then you just take a small aliquot of that and put that into your 96-well plate qPCR mix, and you analyze it. So Yale had a nice sponsorship with the NBA. and their program was called Spit and Swish, which I love that name. And ironically, my collaborators in virology are at Rutgers and UIUC. And in the summer, they sent me pictures of, kind of what was going on. So this is a picture of a spit test. It was one from Rutgers. It was one of the first tests that became emergency use authorized. And, ironically, my collaborator said, oh, in the beginning, it was great. They had this rapid spit test. And then a company called Vault Health took it over. And so then they wanted to make money off of it. So then the test became kind of slow and, difficult to get. But anyway, so they had the first mail-in saliva test, Vault Health. UIUC also does a spit test where you spit into a 50 milliliter tube. It's a pretty big tube to spit into, so it makes it easy. And then those tubes are transported through a golf cart to a centralized testing lab on campus. Okay, so we tried this in my lab. And I just grabbed whoever was working in the lab at the time and asked them to give me a spit sample. I did make them sign consent forms, so it was all legit. And what they did, and including myself, is we spit into a 1 milliliter microcentrifuge tube, and we would just put kind of a regular pipette in there and use that pipette to funnel the saliva down into the tube. So that was pretty fun. I would say not to eat or drink before you do this test. We had someone, I won't say who, it was not me, smoke a cigar before the test. And that saliva was very, very thick, viscous, and difficult to pipette. So that was interesting. This was definitely the first time I've ever tested saliva. So we did the saliva direct method. We combined our saliva with just a small amount of proteinase K, and that breaks up the RNA and DNA nucleases that are in your saliva. So your saliva has a bunch of stuff in it. It's much harder to kind of amplify for the virus in saliva compared to just a regular water background, which is what we would normally do in the lab. So we combined the saliva with this proteinase K to break up those nucleases. We combined them and we mixed them. We heated it up for 5 minutes at 95 degrees, close to boiling, like I said. And then we just heated it up like I said, I've used an oven, a PCR machine, a water bath. You can use any type of heat source for half an hour. So this is kind of what the test looks like by eye. And that's the neat thing about this test, is that I could give you a tube right here. You could spit into it. We could put it into a heater, and you would see your results turn from pink to yellow if you're positive. So this is what we saw. You can see all the tubes were pink to begin with. And then at 30 minutes at 65 degrees, they started to turn yellow. I'll tell you a little bit about my results. It's kind of hard to see because I don't have the pointer, but see if this works. No, does it? Okay, I'll just talk about it. So if you look in the top two, I would say tubes on the very top, that is all in water, right? So water is a much easier background to work with than saliva. So if you look at it, first of all, in the very left column, that's your no template control, because that's water. So if you amplify water, you should get nothing because there's no RNA in there. There's no template. So it's all pink. So that's great. That's kind of a control. If you look at the second column, that's your positive control. So that's where I put in like a little sample that says, okay, yes, this is working. My LAMP assay is working, and it's just a bit of strand of the coronavirus. And so if you look in all of those tubes, they're all yellow, so that's good. That's working. The third sample is an R-actin sample. So it amplifies and is positive if you're a human person. So my lab doesn't do clinical diagnostics, right? So my lab just studies viruses, but we have some variable results there. So if you take a look in water, what do you expect it to be? Do you have a human sample in water? So we actually do not. And so those tubes should all be pink. But you can see some of them are pink and some of them are yellow. So that means our tubes are contaminated with like human sample, which is like everywhere in the lab. And we probably should have kept our tubes a little bit cleaner. And then the last column is whether or not you're positive. So that's the important column. And if you look carefully, if you just have this genomic full-length RNA, which is what I have on the top, so that's the RNA that I extracted from the virus, that's a lot easier to access. Remember I told you have to do that RNA step in the beginning, which kind of purifies everything and gives you pure RNA. Well, that's really easy. You can actually access the, you can see all those tubes are yellow, all the way from 100 copies per microliter all the way up to 10 to the 7 copies per microliter. So in water, whether it's on the left-hand side, I have the full-length RNA, and on the right-hand side, I actually have actual viruses that came from the BSL-3 that have been inactivated. So I'm working in this in my laboratory, but it came from the BSL-3 laboratory into my laboratory. So that's great. At the bottom, you see the saliva, and you can see that for the extracted RNA on the left, the bottom left, that's all yellow, down to 100 copies per microliter. But you can see a pink tube, right, in the very bottom right. Well, that's actually the heat inactivated virus. That's the full capsid virus in saliva. So unfortunately, this test is not sensitive down to 100 copies per microliter, which is why it works as a rapid test, but may not be as sensitive as qPCR. But that's okay. This is just what we got. It's definitely good enough to catch transmissible virus. And other researchers have gotten this assay down to 100 and even lower with some tricks of purification using silica beads. So, like I said, we're probably, like I said, that lowest level was at 100, which we couldn't detect. That's on the left side, that's at the 1. The log 10 of the viral load is at the 1. You can see here my rapid test is exactly where that blue dot is at 10 to the 5. And so that is good enough to see transmissible virus. Oh, you can't see the gray background behind it. But yes, so what they're saying is qPCR is far too sensitive. You know, you can detect all the way down, but it's more important to have a rapid test to quickly detect if you're positive. Okay, my area of research is in microfluidics. So this is a way to really speed up your assay even faster than LAMP, than what I showed you. It's very sensitive and specific because you're working with really small volumes, and it's quite affordable. So it's a lab on a chip, meaning you can do the entire assay in a disposable chip that can fit in the palm of your hand. And that's kind of what I've been working on. So I've been doing all the LAMP stuff to be able to put that assay into a microfluidic chip. These are just some examples. This recently came out in a pretty prestigious journal last week, or at the end of August from UIUC. Stanford Fluidigm is a commercial microfluidic system that has a saliva test. So you can think of it as a computer chip with plumbing. So what it is a chip that fits in the palm of your hand. You're manipulating really small amounts of liquid on that chip. So anywhere between, I would say, like picoliters to nanoliters of liquid in kind of these really tiny tubes that are a little bit like the wires that you would see on a chip, but just imagine them filled with fluid. So here are some examples of microfluidic chips. This is a really common one. It's called, it's from Steve Quake's lab at Stanford. And actually, I should mention that Fluidigm is a company that was founded by Steve Quake back, I think, in the early 2000s. Using this exact chip. So you can see a lot of these chips can be commercialized. My area of research is in drop-based microfluidics, where I'm making these drops. So I have a lot of fun making really fast movies. These drops are being made at 1,000 per second, so you have to take a video of these drops being made and then slow it down to be able to watch what's happening on the chip. So this is a slowed down video, slow it down 1,000 times. You can see drops here. And there's just all sorts of different devices. So paper microfluidics, that's your kind of, you know, you read about these lateral flow assays. Well, that all originated from paper microfluidics, where, you know, you have capillary action moving the liquid on a piece of paper. That makes it, the whole assay really cheap. You have electrowetting, And this is typically what we use in our lab, these PDMS devices, which is kind of an elastomer that's bonded onto glass with these channels built into them. So, like I said, my area of research is in drop-based microfluidics. It's a really specific area of microfluidics, and we make these drops. This video is, again, slowed down 1,000 times. Here you can see actually single host cells for the virus being encapsulated into the drops at very high rates. And what is important here is that we have a surfactant that surrounds each drop, kind of like surfactant you think of soap, right? So these hydrophobic tails and these hydrophilic heads, and on the inside we can put in there biological samples. So cells, proteins, RNA, anything, bacteria we also work on. So anything biological, and it's biocompatible. So we can encapsulate cells. Here's a video of them being encapsulated. There are yeast cells in there. You can collect them all into a small tube. This is a tube that you would actually put into a PCR machine. And in this tube, there's probably about 1 million drops. You can, say, put those drops into an incubator, let your virus grow if they have single cells in there with virus. You can then re-inject them and analyze them. We can add small amounts of liquid to every drop as it passes by. We can do the opposite. We can take off a small amount of drop. And finally, we have the ability to sort. And that was the video if you looked carefully, that was what was happening earlier. But here we can sort drops based upon their fluorescence. So if they have if they're emitting some sort of light when you shine it with a laser, we can sort for those particular drops that are fluorescent and emitting that light. So what am I working on? I guess this was kind of in the middle of the pandemic. And I went for a run and I was like, I want freedom. So I call this the Freedom Chip. It's a cheap, disposable, rapid point-of-care microfluidic device that uses lamp and drops. We're prototyping it now. So this is kind of what you're seeing here on a microscope. And this is kind of the general idea of what I want to do. So I really want the freedom to be able to go to a Bobcat game, to go to a movie, to really like freely go back to work with lots of people. I want you guys to be all sitting here much closer to me. And just to be able to, kind of see family and friends, travel and do the things that we used to always do. And so, and to be able to do this safely, right? So, SARS-1, the one that came out in Asia, it was a lot easier to do this sort of massive screening, because if you had SARS-1, you had a fever. So you could just check somebody's temperature. You could have an IR camera so that when people were walking through airports, you could see whether or not they were infected. So it was really, it worked. It was really fast, simple, effective. However, this temperature screening does not work right for SARS-CoV-2. So that's why we're trying to get all these rapid tests out. So the concept is here, we're going to use saliva. We're going to use LAMP, which I just talked to you about. There's no supply issues with LAMP that you might see with qPCR that everyone is using. It's very rapid, it's selective. We're working on ways to make it more sensitive. Definitely drops will make it more sensitive. So that's the advantage of drops, which I'll talk about. The Freedom Chip here, it's just going to be a simple microfluidic chip. It'll come in a bag with a spit tube, likely. You'll spit into the tube. You'll put that tube onto the chip. The chip will just make drops. This can all be done without a trained operator. We're going to heat the chips up for, probably about 5 minutes. I told you that the assay takes 30 minutes. But the thing is, when you are working with really small volumes and these tiny drops, everything happens much faster, because your effective volume, or your effective volume is just so much smaller. So anyway, we think it's going to take about 5 minutes. We have some preliminary results where it's happening that quickly. And then we're going to read the assay out through a cell phone. So like I said here, It's a little bit different. This particular LAMP assay that I've done here that I'm showing you, I showed you qPCR before. This is LAMP. This is a form of LAMP, not the colorimetric form where you see things going from, yellow to pink, from pink to yellow, which is actually great because you can see it by eye, and I can test you here and you can see. But this is tied to a lab. It's fluorescence-based. But fluorescence-based tends to be much more forgivable with saliva, which has a lot of problems with pH and things like that. And it's also really sensitive, right? So you can detect fluorophores really easily. So we're sticking with, in terms of microfluidics, we're sticking with fluorescence. Like I said, we can really easily measure fluorescence in drops. If you look here, we have the same genes, the E and the RDRP, and they're being amplified. The red curve is really concentrated virus, and then the yellow curve is, much less, down to 4 copies per microliter. And you can see all of these curves amplify within 15 minutes. And we can detect down to, fluorescence is more sensitive than colorimetric, so we can actually detect down to 0.4 copies per microliter, which is great. So it's really, so that's why we're kind of going with an already sensitive method. Just this is in bulk, but then we're going to also put it in drops, which should make it quite good. Okay. We can do the same thing with influenza. It seems to amplify at these concentrations within 10 minutes. And finally, here are kind of is what we're looking at, is we put this into drops. They're amplifying a lot faster than in bulk. So something that's taking maybe about 15 minutes is going down to, I would say, about half that time. So it's much more sensitive and faster in bulk. We can detect, so far we've done about 400 copies per milliliter. That's, your 0.4 copies per microliter. And it's all in one step. So everything is all in that drop, and you just have to heat that drop up. So those are some of our results. Other people are working on this, and you'll start to see some of these come out, I would say, next year, early next year. There's some other methods. They still take, they're just different. There's so many different ways of testing the virus, right? LAMP is one. Another way is sequencing. That's really big. So if you look on the right, all these people are doing sequencing at Penn State. I came from UCLA chemistry, was where I got my PhD. So my colleague there, Shri, is working on a method called SwabSeq, and then LampSeq at the Broad at MIT. But notice a lot of these use LAMP upstream of sequencing, right? Because LAMP is so fast. So no matter what, when you sequence, you have to do this PCR step and then you sequence. So it takes a long time. That's the thing about sequencing. You can send like a self-swab in and get it sequenced, but it takes about 8 hours. It takes a long time. So it's not a rapid test. However, it is very cheap. So sequencing at this point is definitely likely under $1 a test. And then you have kind of these CRISPR-based methods. And I don't know if Blake Wiedenheft here already talked. He is an expert here on campus in CRISPR. But you have both Berkeley and MIT working on these what's called Sherlock at MIT on the East Coast, and a detector on the West Coast. But it's a similar concept. They both use CRISPR. So notice they both use an isothermal method. One of them does use LAMP. I think it's detector that uses LAMP to downstream CRISPR amplification. And then the other one uses a different method called RPA. So really isothermal methods are the way to go because that's a lot faster. But overall, these CRISPR methods still take probably about half an hour to an hour. So that's another method. Okay, with that, I'm really sorry about all the technical difficulties, but I'm happy to have any questions. Feel free to contact me through my email or follow me on Twitter. I'd like to acknowledge the people in my lab and my funding sources. So thank you. Any questions?